The question is that how is pre-messenger RNA splicing carried out? So splicing means the removal of introns from the pre-messenger RNA, and this is carried out through this. It is accomplished by this large complex which is made of protein as well as RNAs. Okay, and this is known as, the whole thing is known as spliceosome. This whole purple ball is known as spliceosome. It is contained of protein as well as RNA. Okay, now it basically, you can see that it attaches itself with the pre-messenger RNA, and these, like the RNA part of spliceosome, it acts as enzyme. Okay, and it cuts it catalyzes the reaction. It cuts the introns from both of sides, and then the exons, they got attached, as you can see here, and the introns are cut out. Okay, the important point to note here is that RNA is acting as an enzyme, and it basically contradicts with the concept, the main biology. There is a very important biological concept that all enzymes are. Yeah, exactly. So this, like the discovery of the RNA as enzyme, it invalidates the idea that all biological catalysts are proteins, because we can see that RNA also act as enzyme, and they have a specific name. Do you remember? Ribozymes. What are the ribozymes? RNA molecules that function as enzymes. What are the ribozymes? RNA molecules that function as enzymes. And I have just explained it, that for example in the spliceosomes, the RNAs, you can see that these small RNAs, they function as the enzymes, so they are ribozymes. Okay, exactly. Okay, now let me check. Okay, now we are going to I told you that the discovery of the ribozymes, it invalidates the idea that all enzymes are proteins. So now let's see what and by the way, not all RNAs are enzymes. Okay, remember that. So just few of them acts as, they functions as enzymes, so they are ribozymes. But remember, not all RNA are ribozymes. Are you getting my point? So we are going to see that what are the properties of the RNA that enable some RNA molecule to function as enzymes.